Good morning. For those of you who do not know me, my name is second class Phil Poller, and I'm an MS-SCP major here at the Academy. After doing research on many different species of tropical marine organisms, I decided to research the zebrafish for my creature feature project. Since the 1960s, the zebrafish, Danio ririo, has become increasingly important to scientific research worldwide. During this presentation, I will discuss general background of the zebrafish, human health issues, and how organisms like the zebrafish are being used to solve many of our global health issues. Let's get started. First, let's start with the introduction. Who are the zebrafish? Zebrafish are a small tropical fish belonging to the minnow family. Zebrafish are commonly found in freshwater streams of the southeastern Himalayan region, areas such as India, Pakistan, Bangladesh, Nepal, etc. They are identified by the 5 uniform blue stripes on either side of their body. Zebrafish typically grow to an average of around 4 centimeters. Their lifespan typically runs around 2 to 3 years, but in ideal conditions can range to over 5 years. Remember these 2 points, as they will be very significant further in this presentation. Now let's get into the problem. For centuries, research has been conducted in attempts to understand the complexity of the human body. As we know, there are hundreds of different diseases and cancers associated with humans. Although progress continues to be made to cure these diseases, there are many out there that have proven to be very problematic, especially in terms of health research. Since we have failed to understand many of these diseases, researchers are having a tough time breaking the code. One example of a human disease being studied is muscular dystrophy. Muscular dystrophy is a group of 30 inherited diseases that can lead to muscle weakness and disability. In muscular dystrophy, abnormal genes interfere with the production of proteins needed to form healthy muscle. The disease affects around 200,000 people a year in the US alone. There are no known cures for muscular dystrophy. However, there is treatment available for those affected. Now let's get into the solution. Good news. Although there are a numerous amount of human health conditions, researchers have begun to find answers they have been looking for centuries. Researchers have found that certain organisms share similar traits, genes, and structures with humans. With the help of model organisms such as rats, mice, and marine fish, scientists have been able to model and trace pathways of different diseases through the use of genes. Back in the late 1960s, the zebrafish began its role as a model organism. A model organism is a non-human organism scientists use to further understand particular biological phenomena with hopes of being able to understand workings of similar organisms. Now let's get into how this program works. Often a patient's DNA is sequenced in order to find a mutation in a gene that could potentially cause his or her disease symptoms. To determine if loss of function of that gene could cause the symptoms seen in the patient, the same gene is mutated or knocked out in zebrafish, and then the fish are examined for similar symptoms. If one or more of the patient's symptoms are observed in the zebrafish knockout or knock-in model, the zebrafish can be used for further studies to help determine why the mutation in that gene causes the disease. Side note. In addition to utilizing zebrafish disease models to characterize human diseases, researchers can also identify and test new drugs to treat the diseases being modeled. In other words, in addition to tracing the cause of each disease, researchers can also use zebrafish to test new medical technologies to better treat the diseases already found. At the top of the page, you can see a laboratory. In this laboratory, eggs are kept and stored on shelves with constantly flowing water on the left-hand side of the image. In the middle of the image, you can see a shelf with different labels, pipes, and holding bins. It is here that each embryo, juvenile, and adult phases are studied. Now let's get into the benefits of using zebrafish to study human health and disease. As I have mentioned earlier in this presentation, mice have been used for years in order to study human health. Although mice are evolutionarily more similar to humans because they are mammals, zebrafish have several advantages over mice. One important advantage of zebrafish is that the adults are small and prefer to be housed in large groups or shoals. As a result, they require much less space and are cheaper to maintain than mice. Another advantage is the adult zebrafish breed readily, approximately every 10 days, and can produce as many as 50 to 300 eggs at a time. This is quite different from mice, as they generally produce litters of 1 to 10 pups and can only bear approximately 3 litters in their lifetime. Scientists' experiments are generally repeated multiple times in order to prove that the results are accurate. So having an animal that can produce a large number of offspring over and over is very helpful. Another important point is that zebrafish embryos are also laid and fertilized externally, which allows them to be easily manipulated in a variety of different ways. Zebrafish can grow at an extremely fast rate, developing as much in a day as a human embryo can develop in a month. Zebrafish embryos are nearly transparent, which allows researchers to easily examine the development of internal structures. Every blood vessel in a living zebrafish embryo can be seen by just using a low-powered microscope. Zebrafish have a similar genetic structure to humans. They share 70% of genes with us. 84% of genes known to be associated with human disease have zebrafish counterpart. As a vertebrate, the zebrafish have the same major organ tissues as humans. Their muscle, blood, kidneys, and eyes share many features that we humans share. Another interesting point is that zebrafish have the unique ability to repair heart muscles. For example, if part of their heart is removed, they can grow it back in a matter of weeks. Scientists are working to find out the specific factors involved in this process to see if it will help us to develop ways of repairing the heart in humans with heart failure who have severed heart attacks. Lastly, the zebrafish genome has been fully sequenced to a very high quality. This has enabled scientists to create mutations in more than 14,000 genes to study their function. As I have stated earlier in this presentation, the study of genes in the zebrafish through the knockout and knock-in program has been shown to closely resemble the progression of the human disease muscular dystrophy. Patients with muscular dystrophy have been found to carry mutations in dystrophin and demonstrate childhood muscle weakness that gets progressively worse. In both humans and the zebrafish model, the loss of this dystrophin gradually leads to necrotic muscle fibers that are replaced by inflammatory cells, fibrosis, and abnormally sized muscle fibers. As you can see in the figure, this figure shows the visual differences in muscles between wild-type zebrafish larvae and dystrophic larvae. As you can see in the figure, the difference between a healthy zebrafish and a zebrafish that struggles from dystrophic muscle failure is quite severe. The significance of the zebrafish. Zebrafish have already been used to help unlock a number of biological processes behind muscular dystrophy and are an important model for understanding the mechanisms of development in diseases such as cancer, melanoma, and even heart disease. It's crazy to think that such a small organism can play such a huge role in human health. Without model organisms like the zebrafish, many of the technologies, medicines, and advancements in human health that we've reached today may not be possible. Thank you, and that concludes my presentation.